Throughout the second half of the 20th century, spectacular technologies revolutionized scientific understanding of the cell, the basic unit of life. During an interview with biochemist Michael Behe, Stobel learned how this new knowledge has shaken the foundations of Darwin's theory.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all. But with the hard work of science in the 20th century, we've seen that the cell is far from simple. It's got very complicated molecular machines and things that are very resistant to Darwinian explanation.
Michael Behe has devoted his career to the study of the design and operation of the cell. He has also written extensively on the biochemical challenge to evolution.
Most people have no idea of how small and complex cells are. A typical cell from you or me, called a eukaryotic cell, is probably 1/10 of the size of the head of a pin. And yet in that single cell, there are about 3 billion units of DNA making out the chromosomes. And those 3 billion units make the molecular machines of the cell, literally machines that make the cell work.
With computer animation, we can enter the cell. Here, the staggering complexity of its molecular machinery is clearly seen.
It's like going into an automobile factory. The factory has a large number of machines. The parts have to fit together in very specific ways to do their jobs, and if things go wrong, the cell is in big trouble. And just one cell is enormously complex, but humans, you and I, are made from trillions of cells, and those trillions of cells have to fit together in the right way and do their own job. Darwinism was a lot more plausible when we were thinking about globs of protoplasm than it is when we're thinking about molecular machines.
Each of these biochemical machines is a masterpiece of engineering and nanotechnology. They are essential to functions as vital and diverse as vision, photosynthesis, and the production of energy in the cell. Michael Behe has studied several of these machines, including the flagellum, a remarkable rotary motor.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It's had a propeller and the hook region and the drive shaft and the motor. And I looked at that and I said, that's an outboard motor. That's designed, you know, that's no chance assemblage of parts.
Behe's reaction was not surprising, especially when the bacterial flagellar motor is animated and magnified more than 50,000 times to display the details of its construction and operation.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
It's got some tail proteins which act as the propeller. When the flagellum rotates, these push against the water and therefore push the bacterium forward. And the motor uses a flow of acid from outside of the cell to the inside of the cell to power the turning.
The bacterial flagellum has 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller. It's not convenient that we give them these names. That's truly their function.
In all, about 40 different protein parts are required to build a flagellar motor. Half of them are constructor proteins, specialized mechanisms that assemble the flagellum's individual components. Since its discovery, biologists have tried to understand how a machine of such superb design could have arisen gradually, without foresight or plan, through the biological pathway Darwin envisioned.
I think what Darwin was trying to show was that things that look designed aren't really designed, but that we can find naturalistic processes to account for the complexity of life. Darwin theorized that every part of every living organism evolved through natural selection, a blind process that acts upon random changes in the cell.
Darwin believed that given enough time, these random variations would transform the simplest cells into the great diversity of life that inhabits our planet. In his study of evolution and molecular machines, Michael Behe has raised a significant challenge to the creative power of Darwin's mechanism of natural selection. It is called irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
Irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function. Catching mice. The concept of irreducible complexity also applies to biological machines, including the bacterial flagellum.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell. You can't put something like that together gradually because they need a large number of parts interacting with each other at the same time before they work at all.
Without the tools to observe the machinery of the cell, and long before the idea of irreducible complexity, Charles Darwin offered a way to test his own theory. In Origin of Species, he wrote, "If it could be demonstrated that any complex organ existed, which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down."
Darwin acknowledged that if someone identified a biological system that could not have been constructed in incremental steps over long periods of time, then his theory would be invalid. And what Michael Behe and others have discovered is the existence of biological machinery that cannot be explained away by Darwinian processes. Darwin's failed predictions have in fact falsified his own theory.